Hey everyone. Well, primarily Professor Booker. My name is Caden Bratcher, and today I'm going to walk you through my 3D model of the protein that I will be presenting, which is called melanocortin 1 receptor, or Mc1r for short. Now, it sounds a little complicated, but I'm kind of going to explain what it does. It's a receptor, which means its job is to sit on the surface of our skin cells and wait for a specific chemical signal. So that signal is a hormone that tells the cell to make more melanin, the pigment of our skin and our hair color, and also protects us from damaging UV rays like the sun. So basically, when you're out in the sun, your body sends that signal to Mc1r, and Mc1r tells skin cells to get to work, making melanin. And this helps protect your DNA from getting fried by UV rays. So the mutation I was assigned is called ASP294HIS, which means in the normal version of the protein, there's an amino acid called aspartic acid at position 294. And in the mutated version, it's replaced with another amino acid called histidine. I hope I'm pronouncing that right. It might seem like a tiny change, but it can mess up everything, basically. So I basically want to go over what it's looking like now. I had to work with what I had. Okay, no need to judge. I feel the judgment. I know you see the tape. Okay, so the overall shape of this is kind of like a twisted bundle of tubes. The tubes are called alpha helices, and there are 7 of them. They go through the cell membrane and help the protein stay in place while it receives signals. So to build my model, I used pipe cleaners and to represent those helices because they're flexible and easy to shape, as I did so well, as you can see. And, I also use straw and connectors. See, I got the straw and the connectors as the tape to represent the loops between the helices that help the protein fold into its full shape. Now, I actually made 2 versions. Okay, so here's the original and then the mutated. On the normal version, I used more of a blue to represent like the amino acid, aspartic acid, and I used the green to represent the histidine. And to show how the mutation affects the shape, I slightly bent the helix differently in the mutated model, which also is a lot more obvious because you can see the mutation in this model when you see there's blue where the yellow is supposed to be. Now, the charge and size of the amino acids, which can mess up how the protein folds, and it shows how it works. So if the normal protein, the signal is received properly and the skin makes enough melanin to protect against the sun, but in the mutated version, the signal doesn't go to the melanin and protect against the sun. But in the mutated version, the protein becomes less active, which means the cell makes less melanin, which is why I made this part lighter than this one, just to show a bit of skin difference. This can lead to lighter skin, of course, red hair, freckles, and more sensitive to sunlight, like albino people. You know what I'm saying? Like, there can be black people. But it can raise the risk of skin cancer too. Now, is the model perfect? Not really. There are some things it doesn't show. First of all, the real protein is way smaller than anything I could build. And the shapes I use, like the pipe cleaners and beads, are just a rough way to show the structure. Also, the protein doesn't just sit still like this. They're always moving, interacting with other molecules and reacting to the environment. But my model doesn't include the cell membrane or the watery surroundings where the protein would actually live. But even with those limitations, it's a helpful way to see where the mutation is and how it affects the protein. So to wrap that up, that is my model of Mc1r and how that ASP294 HIS mutation affects it. I hope it helps you understand how a single amino acid can change a whole effect in the protein and even something as big as skin color. Thanks for watching, y'all.